I've got to apologise. I forgot to introduce myself at the beginning of the session. My name's Kerry. I'm from the Faculty of Health. I'm being your chair this afternoon. And thank you for that, Nigel. Okay, next presentation is Roshni Paul, Anaerobic, Digestion and Microalgae. Thank you. Did I say that right?
It's all right. Yeah.
Thank you.
Thank you. Can you all hear me? Excuse me, can you all hear me? Yeah. Okay. I'm Roshni. I'm from the Faculty of TE and from Centre for Low Carbon Research. It's quite difficult to talk about my presentation soon after lunch, and I'm glad that you, Nigel, you did it before mine because it gave me some time to relax. Well, I gave the presentation title to be Anaerobic Digestion of Microalgae, and then I changed my presentation to Our Mad World, because my research is all about madness. Not truly madness, but about MAD. So shall we talk about some madness? Right, so these are my objectives for today. It's all about renewable energy from microalgae, and then obviously I talk about MAD, and then what's the processes and the requirements, and what are the challenges that I've been facing through the entire research, and then what are the solutions that I have identified through this research. You've all been hearing about renewable energy. It's always in the news or in your day-to-day lives and everything. So anything except from the fossil fuels, you name it as renewable energy. And why microalgae, and why not solar or wind or all the other different types of renewable energy that's already available in the market? Now, if I talk about the creation story, microalgae comes for me first. So they are recognized as one of the oldest living organisms in the world, and they are very fastly growing microorganisms, because it's 100 times faster than that of a normal plant. And one of the interesting factor of microalgae is the accumulation of the lipids, which can be used for the derivation of biofuels, one of the valuable products that you get from microalgae. And my research area is concentrated at the source of biomass. So how do you make use of the biomass, which is after the derivation of biofuels out of the microalgae? And also, when I talk about microalgae, you've already heard about renewable energy and biofuels which are being derived from corn or yeast or like soybean or jatropha. So they all come under the second or the first generation of biofuels, and microalgae comes in the third generation of biofuels. Microalgae has also got the carbon sequestration potential. It's a long word, but it's just that it has the potential to capture carbon dioxide and make it a less carbon-free world. This is a diagram, which is a conceptual diagram for microalgal biotechnology. If you start from the left-hand side of yours, microalgae can be grown in an open pond or in like long circular tubes, which are called the photobioreactors. You cultivate them, you put the nutrients, and you provide the carbon dioxide and the light, which is artificial or the sunlight, and then you harvest it, and then comes the biofuel recovery stage, and the biomass which is left out of that you put into anaerobic digestion, which can be used for power generation and combined heat and power. And from there, the carbon dioxide can be resupplied to the cultivation pond, so the cycle goes on. So the entire system is carbon neutral in this way. Now, what is MAD? This is my MAD world. MAD stands for microalgae anaerobic digestion. Now, anaerobic digestion is nothing, but it's a natural process which is the breaking down of the biomass into simpler organic matter from which it was naturally formed off. And here, my biomass source is microalgae, and hence the name MAD. And biomass is any material, excluding fossil fuel, and it can be used as a, and any living organism, which in due course can be used as a fuel, either directly or indirectly by a process. One of the major products that you get as a result of anaerobic digestion is biogas, which is a clean fuel, and it's a mixture of methane and carbon dioxide. Again, you're using up that carbon dioxide to grow microalgae and then to make the system carbon neutral. Once you've taken the biogas out of the biomass, the substance that is resulting is called the digestate, which can be converted into either a fiber and also into a liquor. It's one of the problems of anaerobic digestion that people are facing these days, how to make use of digestate, because normally they are being used as soil fertilizers or conditioners and then chucked back into the agricultural fields. And the liquor, which is being separated from the anaerobic digestate, is just used either for the cultivation of microalgae so that the system can be better utilized. Again, why you are doing MAD? It forms as a connecting link in this renewable energy technology, because whenever you are talking about microalgae, first of the things that comes into your mind is about the cost. How do you manage to separate the lipids out of it? It's a costly process. Well, you separate the lipids out of the microalgae, and then if you are employing anaerobic digestion to make utilize of the biomass, then in actual fact it can provide as a connecting link and then balance the costs. And also there's another option for co-digestion, which is also one of the areas of my research. It is also the identification of other co-digestates, like the other digestates which can be digested along with microalgae and then make it a bit more renewable. This is similar to the other picture that I showed you, but it's more specifically about AD, and then you can see that the biogas which is produced is directly used for CHPs. It's a flexible technology, really. It's in about the size and also where you can produce the plants for AD. It can be as small as a room, or it can be as big as because Severn Water utilizes the AD technology. So it's been an already established process in the UK and elsewhere, but it's not being quite used for microalgae. So that's where the research gap is. The process as such, it occurs in 4 different steps, but it's very simple to control every single step. So that's why it becomes more controlled. We call it anaerobic because it happens in the absence of oxygen. It can be also varied in terms of the temperature that it can be controlled. If it's under 40 degrees Celsius, then it's called a thermophilic anaerobic digestion, and if it's above 60 degrees, it's called a mesophilic. As I already told you, it can be several stages. So if the digestion occurs in just one stage, it's called a single stage digestion, or if you're incorporating more than one stage, it's called a multi-stage one. Another term that's been included newly is called the pre-treatment. Well, after the derivation of the biofuels out of the microalgae, you can actually pre-treat the biomass so that it can be digested a bit more efficiently to produce a bit more biogas. Literature review shows that pre-treatment actually increases the biogas yield, and also it breaks down the biomatter quite easily, so that if you are using it as a fertilizer, it can be degraded quite easily. It's simply the repetition of the same thing, and one of the factors that I would stress here is about waste to energy. Today's world, we know that we are generating a lot more waste, and if you're incorporating AD technology, then it can be one of the useful ways by which you can generate energy out of the waste. This is our vision. It's called a biorefinery. A biorefinery is a place where you integrate multi-processes to produce biofuels and byproducts and other valuable products out of it. So my project is the AD bit, and one of the students, Jim, he's looking at the cell disruption, which is a pre-treatment, and another student who's Tim James, he's looking at the growing of microalgae at the photobioreactor at the farther end corner. This is our lab, so time for some photographs. So this is microalgae. You are familiar with the name spirulina, I guess. So that's the algae there. And when you look in through the microscope, you can see these cells, and you can actually see the cells, they're splitting up. This is some of the snaps of the work that I have done. So those are the cells which are before pre-treatment. And if you simply crush the algae with a mesh lamp water, that's the image that I've got there. And then this is another process of pre-treatment, which is autoclaving. And you can see that up there, the cell size are not quite uniform, but when you're using autoclaving, the cell size is very uniform. What are the challenges being identified? I would stress at the development of different pre-treatment methods here. The other ones are the overall AD-specific challenges. And my research is basically looking at the different pre-treatment methods. I'm looking at ultrasound, because ultrasound is being, literature shows that ultrasound not only disrupts the cell, but the cell wall and the membrane, so that the matter can come out quite easily, so that it would help in the biofuel derivation, and then the biomatter can be used more for AD digestibility. And I'm also looking at microwave, autoclaving, and the simple mechanical methods. Okay, yeah. Overall, these are the R&D categories. As I explained earlier, these are my solutions that I have identified. Cell wall is the main barrier when you come to a microalgae, and then simpler and economic methods have to be developed if you have to employ it in a large-scale one. And then co-digestion, I have identified 3 different feedstock, which is brewery spent grain, agricultural waste, and sewage sludge. And again, literature review has shown that this will increase the biogas yield. Sorry, thank you. I had to wrap it up quite. I know it's quite a lot to take it in, and it's not, because I might take the whole day and take a lecture on this if you are ready, but sorry, and I hope you have enjoyed it. So this is our mad world. That's my supervisor, Dr. Lindsay, up there. That was my cooler supervisor, Dr. Mike Zulu. He has left me, but he's still my supervisor, and that's me there. Thank you for listening to me. Peace.